So I'm going to talk to you about a notion of memory, a view of memory as a fundamental aspect of living organisms that can occur at very, very different levels. I'm going to talk to you about a historical figure that many people are not very well acquainted with, Richard Semon, who is one of my own heroes, and he's one of the people who thought most deeply in the beginning of the 20th century about memory. So first of all, I want to thank my colleagues, Marion Lamb and Zohar Bronfman and Simona Ginsburg, with whom I discussed many of these ideas and also wrote about some of them. And this lecture is dedicated to Marion Lamb. Now, the interesting thing about memory is that it is that there was a very broad view of memory as a process that unites organisms present with its past both during the organism's lifetime and across generations during phylogeny. And this kind of view began to be developed around the 1870s, in the 19th century. And it was based on analogies that people found between learning and memory and heredity and evolution. And one of the people who expressed this kind of notion relatively early on was Samuel Butler. He said, "Life is that property of matter whereby it can remember. Matter that can remember is living. Matter that cannot remember is dead. Life then is memory." For him, anything that shows memory shows indication of memory, it must be something alive. And according to all this view that were developed by several people since the 1870s, the inheritance of acquired character was assumed to be in place. It was a necessary assumption within this tradition. Now, what were the analogies they were thinking about between ontogenetic memory, physiological and behavioral cognitive memory, as we would put it today, and heredity and evolution? Why did they think that there must be some kind of common denominator? First of all, they were thinking about repetition that is leading to memorization, which we see in mental memory. When we memorize something, repeat it again and again, well, this is something that we remember. And then they also claim that repeated conditions lead to transition from acquired to innate characters. And many different people who you can see the list here thought in this way. Then there is some kind of orderly procession in memory. So skilled behavior, for example, there's one thing that is going up, a sequence of one thing going after the other. And the same kind of thing you can see in embryological progression. There's also one thing going after another, and in a sense also leading to another, to something else. And recapitulation was thought to be the heredity evolution analog of this. And that was especially Ernst Haeckel who pressed this point. Another thing was condensation. For example, in habit formation, when we form habits, we can sort of skip some stages of what we've learned before, so it becomes much shorter, much more efficient, the habit. And in phylogeny, again, recapitulation, according to Haeckel, was showing the same thing. There was retrieval through partial induction, which is something that Richard Semon was stressing. So we see retrieval following learning. A cue, a very partial cue, can lead to retrieval of a memory. And he claimed that all innate characters are in fact context sensitive. So some kind of change, small change in context, can lead to induction, and therefore to some kind of retrieval at the developmental level, and in the evolution at the evolutionary dimension. Then there was habit formation, automatization, and the parallel was instinct. There were recency effects. In memorization, we remember the recent things. Or sometimes when one has Alzheimer's, one forgets the recent thing and remembers and only old memories are retrievable. And in evolution, we have recent additions that tend to be terminal and unstable, and this was emphasized by many other people. And there's something called reversion, something with people talk in some pathological cases, but also in normal life. There's something like capricious memory, And in pathological cases, like old memories are the ones that are recurring again and again. And the analog, in evolutionary analog, was atavism. Now, the person who made most of this kind of analogs and the person who developed a model of models and discussed in depth the necessity for thinking about a common denominator was Richard Semon. And he wrote in 1901, he was thinking about cell memory, and he wrote in his diary an attempt to simplify the problem of biology. So he was thinking not only about neural memory, but about cell memory, because he was a zoologist. He was trained. He was one of the star students of Haeckel. There's a very interesting story about his life, which I will not go to. But he recognized the similarity between induction and developmental induction and retrieval during the process of remembering. He wrote two major books on memory. One was the mneme, which was translated in 1921. You can find them online. And the other was and that was about mainly cellular memory, memory in plants, memory in non-neural organisms, memory everywhere. And the other one was the mnemischen Empfindungen, which was translated as mnemic psychology, which focused on neural memory. Now, Semon, unfortunately, was depressed over the neglect of his work, was troubled by Germany's status following the World War, and shattered by his wife's death from cancer. And he took his own life on December 1918, unfortunately. He was a very great theoretician. Now, his notion of the mneme, of the principle of memory, mneme is the Greek word for memory or remembrance, and it describes an organism's ability to conserve the facts of stimulation and to interact with the environment on the basis of past conserved effects. This was how he thought about memory in a very, very general way. And he used the term memory not only for storage, but it encompassed for him both latency, storage, and recall. Hence, his notion of memory was basically also a notion of learning. So he developed several terms, some of which are terms that we use all the time, like engram, a memory trace, which is the result of storing some of the effect of the encoding of information into memory, and this he called engraphy. And he was talking about ekphory, the retrieval process. And the reason that he invented all these terms was simply because he said that the existing terms are too laden with association of neural memory, the kind of memory we are used to, and therefore we need some kind of terms that are different, that can be used in a much more general way and do not carry all the associations necessarily of neural memory, although of course there are parallels. Now, he assumed, like all of these biologists and psychologists who were thinking about heredity as a form of memory, that the effects of cellular memory can be inherited. And there were many, many mechanisms that were suggested by many people, including Francis Darwin, Darwin's son. For example, they were thinking about transmissible electrical patterns, about transmissible vibration, about small molecules that were crossing the Weissman barrier. They didn't know, of course, about RNA or they knew something about proteins or something like proteins, but they were thinking very much about some molecules that were passing and leading to these memory effects. And there were actually many, many observations of transgenerational inheritance which seemed to suggest that something like such processes can occur not only between cells during ontogeny, but also between generations. The problem was that these were very messy. The results were often messy because sometimes it worked, sometimes it didn't work. It was very, very sensitive to conditions. And if people didn't repeat the experiment in a very precise way, it didn't work, then it did work. So the that was one problem. The other problem was that there was really not good mechanistic explanation of how ontogenetically acquired characters or memories can become inherited between generations. They didn't have a very good model. And the other thing was that there was an incredible opposition, formidable resistance by August Weismann and the Neo-Darwinians, who explained inherited developmental effects as a result of parallel induction, so that you have induction both in the soma and the germline, not transmission from the soma to germline, but in parallel, both of them in parallel are induced. Second, direct effect on the germline, so the soma is not induced, only the germline is induced, and a selection of cryptic variation that you can't see, all kind of things that are unraveled as a result of recombination, for example, and infection, viruses. And they could explain the transgenerational effect by interpreting them through this mechanism. This, of course, doesn't mean that the mechanism suggested by Semon and the other people who were thinking about the transmission of molecules or vibrations or electrical patterns were wrong. But because it was not acceptable, because they couldn't prove their point, the other mechanisms were thought to be much more plausible. And the result was that Semon's ideas were neglected for most of the 20th century. What was recognized was his focus on retrieval during learning, neural learning. That was recognized by people like Daniel Schachter and Elvin Tulving, and also others. But the other aspect, the aspect of the importance of cell memory and non-neural kind of memory systems, was almost completely forgotten. And it's now, his term engram did catch, that was the only term that did catch, but his general approach to cell memory and heredity was neglected with some because of the Lamarckian overturns, mainly, I think. And there were some people who did recognize the importance of his kind of approach, and one of them was Erwin Schrödinger, who regretted that the physiological model of Semon's theory of memory had not yet been developed, and he wrote, "Important though it would be for the advancement of our knowledge." And there were other people too, like Russell, who thought that this was important. Now, what do we know now about non-neural memory systems? We know about epigenetic mechanisms of cell memory and cell heredity, and we know that there are several such mechanisms. Some are based on modifications of DNA, for example, DNA methylations, and some are based on modification, various chemical modifications, many different modifications of histones, around which the DNA is bound. We have also autocatalytic kind of systems that lead to the recurrence of the same state through positive feedback mechanisms. We have, for example, this is the simplest case, we have templating of the same kind of state of the protein complexes or complex proteins through three-dimensional templating, like in the case of primes. And we have something like we can almost call memory molecules, small RNAs that can be amplified in several different ways and lead to and can be maintained within cells and lead to certain physiological behavioral states which are sustained over time. Now. We have learned during during the last part of the 20th century and during the last 23 years that transgenerational epigenetic inheritance, something that they of course didn't call this way, but some kind of transgenerational inheritance, is actually ubiquitous. And we have many examples of this in single cells, for example in yeast, in plants, in worms, in arthropods like Drosophila and Daphnia, in mammals, and we know that there are differences between in the kind of mechanisms that are employed. Usually, it's more than one mechanism, And there are differences also in the fidelity of transmission. But we know now that epigenetic inheritance is just everywhere. So the fact of some kind of trace of memory of learned or acquired characters that are passed on to the next generation is now generally accepted, although the evolutionary implications of this are still hotly debated. Now, I want to say a few words about chromatin, about epigenetic learning in single cells, not just memory, but learning. So, if we are thinking about chromatin marks, for example, DNA methylation, we must, in order to think about it as learning, not just transmission of something that occurs and is transmitted faithfully from one generation to the next, but about some form of learning when there is also latency and retrieval, then we must assume that chromatin marks, for example, may decay or accumulate with time, that there is persistence of a partial pattern of chromatin marks, and the partial pattern does not lead to phenotypic output in the absence of the stimulus, but the threshold of the reaction is altered. And single gene and gene networks may be involved in this kind of process. And I'm talking about single genes and gene networks in single cells. And here are very, very small models. The first is not learning, it's just storage and transmission. So we have an input, the input changes chromatin, there are the little pluses there, and we have an output, and we have some kind of output, phenotypic output, and over the generations, what we see is that the changes in chromatin are transmitted faithfully, so we have, in every generation, we have the same output. However, we can also have something like memory with linear decay. For example, we have an input, and then we have an output, and then there is a gradual decay over generations of the chromatin marks, so there are less and less of them, and the input also is output also is smaller and smaller. And we can have decay with the threshold. So if we have an input, we have it can be partially transmitted, but leads to the same effect. And then when a certain threshold is crossed, when the decay is there are very few marks remaining, we have absolutely no output. Now, we can also imagine that what we need is actually accumulation of epigenetic marks in order to have an input. So we can have, and so that we need a repeated induction, basically, so that we have an input and we put some epigenetic marks there, and then another input, and only when we have enough epigenetic marks shall we see the output. So not everything will happen immediately, right? It is very, very likely, according to many, according also to Semon, that this kind of process, the accumulation of epigenetic marks over generations may be important for a persistent change in phenotype. Now we can also have we can have some memory with cumulative marking and assimilation. This is and so we can sort of have a first input and it is internalized, and then a second input, and then from then on, that's it. It's like it's going to be very stable, and no input is anymore required. We don't need an input here. This is similar to some kind of genetic assimilation. It's epigenetic assimilation, of course. But we also can have models of very simple non-associative learning, we can also have simple models of associative learning, actually, but here we can develop toy models of simple non-associative epigenetic learning, like sensitization and habituation. With sensitization, we have the decrease of the response threshold, which leads to increased response to an original stimulation, and to response also to very partial stimulation. And in habituation, it is the opposite. We increase the response threshold. So here we have a first case of epigenetic of sensitization. It's again a very simple toy model. So we have an input. Some of the input is internalized. We don't see without an input. We don't see anything. There's no output. But since these marks have been internalized, now the same input will lead to a greater output. Or we can have another case where we have an input, part of it is internalized, and after that, and there is no output, and then, but because there is internalization of some epigenetic marks, there is a very small input can lead to the same output. So and we can have more complicated models. With habituation, there must be inhibitory modification in place, so that when the output leads, the output itself leads to the production, to the generation of inhibitory marks. Not excitatory marks as before, but inhibitory marks. And we can imagine very simple other cases of associative learning, which will require much more complicated assumption, but not very complicated assumption. And we came therefore, and these models were developed by Semon and myself years ago, and we came with the notion of an epigenetic engram, a cellular structure or activity that acts as a memory trace, and is a remnant or specific modification of an original induced epigenetic mark or structure. And it can persist for a long time in non-dividing cells, or it can be transmitted during cell division through mitosis and or meiosis in eukaryotes. And what it does is it facilitates the reconstruction of the original phenotype, the phenotypic response, upon a subsequent induction in the next cell or in the next organism generation, depending on what we're looking on the system. And we suggested there is something that we can call epigenetic recall, the facilitated reconstructions of previously induced phenotypic responses based on the persistent epigenetic engrams. And we also suggested that this kind of mechanisms, based on the different epigenetic inheritance memory and inheritance system that we recognize today, that we begin to understand better and better at the biochemical level, can underlie simple learning in protists, for example in paramecium during ontogeny and also following reproduction. And that such epigenetic mechanism will also be found in simple learning in plants and other non-neural multicellular organisms. We'll have to add here, of course, more assumptions about the transmission of this mechanism of memory traces from somatics between cells, but it is not difficult to think about it and to model it. So this is about single cells. What about neurons? How is epigenetic learning involved in neural learning? Because after all, neurons are cells, so they must have, and we know that they do have, all the epigenetic memory mechanisms that I was talking about. There is cell memory in neurons. What do we know about it? So, first of all, many studies show that there are causal relations between changes in epigenetic chromatin marks in neurons and memory learning processes at the synaptic and behavioral level. And this usually occurs in a subset of neural cells in the network, which are called engram cells. And there is also transfer of molecules, we know, that modify memory via extracellular vesicles. So, synaptic memory and nuclear memory are codependent and seem to interact. This is the result of the last, I would say, 20 years of studying epigenetic memory in neurons and in behavior, mainly in mammals, but not only. And it seems that these mechanisms, that nuclear memory and synaptic memory operate at different time scales, with epigenetic memory being more long-lasting than synaptic memory. Epigenetic and chromatin patterns are involved not just in storage, but also in acquisition, consolidation, and recall process. So there is always kind of two-tier learning. Learning is not just, we have learning at the level of both the epigenetic level and the synaptic level. And they interact they can be also decoupled. And I want to say that I'm talking about epigenetic changes, but DNA changes may be involved too, especially during learning under acute or chronic stress, when we have some evidence that sometimes we have actually changes in DNA, in the number, for example, of repeats, DNA repeats. So maybe there is three-tier memory, and there are also bioelectric fields that hold large-scale patterns together. Not only large scale, but also small scale at many different scales. And the question is, how are these involved in behavioral memory? Do we have a four-tier memory? we obviously have to think about memory at very, very different levels of biological organization. Now, what we do know about memory effects in neuron is that we have gene-specific effects, and we also have global effects. At the level of specific DNA sequences, DNA methylation in particular sites may increase or decrease during learning. The global effect, in the case of global effects, what we know is that improved learning is positively correlated with global increase in histone acetylation and a decrease in DNA methylation. And we also know that other epigenetic factors that enhance DNA methylation also enhance learning, for example, some regulatory RNAs. We also know that things that happen in one neuron, changes in the epigenetic state of one neuron, can be transmitted to another through vesicles in which contain either regulatory RNAs or DNA binding proteins, and they can be transmitted from neuron to neuron, from neuron to astrocyte, and from neurons from neuron to non-neuronal cells. And they can do, and this kind of transmission is very important because it enables synaptic specific inhibition. It allows alliance and stabilization among communicated neurons that can lead to coordination and long-term stability within the network. We can think about coordinating neuronal and somatic activities, and we can think about transmission of the contents of these vesicles to gametes that can lead to the inheritance of behavioral learning dispositions. So now, how can we think about it? I created this diagram because I was thinking. Yes.
I think we're going to have to wind up now.
Okay.
We have to
so
have
I just want to say.
talk.
Okay, fine.
Yeah.
So I will just finish with this figure, which was based basically on the figure of the relationship between DNA, RNA, and protein. Here we have a relationship between chromatin, non-coding RNA, and protein. And what we see here that all at each of this level that there can be self-sustaining mechanisms, and there are interactions which are going both ways, unlike in the case of DNA, RNA, protein, that we don't have a direct, we have indirect, but we don't have a direct connection between the RNA, the protein and the RNA or the DNA, but here we do, because we're not talking about DNA, we're talking about chromatin. So in this model, it's a two-way mechanism. Unfortunately, I don't have time to go to my last slide is that I think what we have learned is vindicating Semon's ideas. That first of all, epigenetic memory in cells is a fact. Epigenetic mechanisms are central to neuroplasticity, where memory is reconstructed. It's a reconstruction process, not a replication process. Epigenetic inheritance occurs across generation and enables the transmission of developmentally acquired variation, including learning dispositions. And the engram, I think, needs to be studied at several levels. Thank you very much.
Okay, thank you very much, Eva. I'm glad we were able to give your talk. Unfortunately, I'm afraid we don't have time for questions. We've got to move on to the next speaker, so thank you again.